Hello students, this session is very important. I am just giving the flow chart of the process of transcription in comparative manner, that is the requirement of transcription in prokaryotic cell and requirement of transcription in eukaryotic cell. Before we summarize and differentiate prokaryotic and eukaryotic transcription, let us begin to learn what exactly transcription is. It is a heterocatalytic function of DNA, where the set of information in the form of nucleotide sequence representing a transcription unit we call gene. Now a specific gene is present in a segment of DNA, and that segment of DNA, when it unwinds, recall yesterday's diagram of transcription unit. When unwinding of DNA occur, the two strands are separated. There is no hydrogen bonding. The strand from 5' to 3' is said to be coding, and the complementary strand is said to be template. So during transcription, the flow of information is given by DNA template. Template is 3' to 5'. The flow of genetic information is copied from DNA template to RNA. So, in other words, transcription is the synthesis of RNA molecule on DNA template in presence of an enzyme called RNA polymerase. RNA polymerase require a binding site, and that binding site is present towards 5' end of coding strand. Coding strand is 5' to 3', and that coding strand is called upcoming strand or upcoming point. It is specified by promoter gene. So RNA polymerase is recognizing promoter gene, and then transcription begins. Towards 3' end of coding strand, there is another nucleotide is present called as terminator region. It specifies that need to stop the synthesis of RNA molecule. Now for this RNA production, what exactly requirements are? Just read the list given in the center. A part of DNA template, not the entire DNA template, a part of DNA template. Enzyme necessary is RNA polymerase. Now here remember, in prokaryotes there is only one RNA polymerase. Maybe production of mRNA, rRNA or tRNA. But in eukaryotes there are 3 different types of RNA polymerases. It is known as polymorphism at RNA polymerase activity. I have specified here. If there is a production of rRNA, then which enzyme is necessary? RNA polymerase 1. Whenever there is a synthesis of mRNA is going on, RNA polymerase 2 will catalyze the activity. And for the production of tRNA, RNA polymerase 3. Hope you understand the first difference in prokaryotic transcription and eukaryotic transcription. In prokaryotic transcription, it may be any RNA production. Catalyst is only one, RNA polymerase. In eukaryotes, depending upon the type of RNA molecule, polymerase 1, 2, 3 are available. This is showing polymorphism at the genetic level. Ribonucleotides are required. Now what are ribonucleotides? Adenosine monophosphate, adenosine guanosine monophosphate, cytidine monophosphate and uridine monophosphate. Here thymine is not required for the production of RNA molecules. Energy is required in the form of ATP molecules. There are elongation factors, there are initiation factors, elongation factors and terminator factors. These all are vitamins along with mineral salts regulating the enzymatic activity. So these are certain requirements. It may be any transcription in prokaryotes or in eukaryotes. Let us learn just steps in transcription in prokaryotes. You can see unwinded DNA now. Here, after unwinding, we can see two strands shown in green color. 5' to 3' is the coding and 3' to 5' is the template. Near the template, the set of information is already flowed. It is shown in pink color. Now, if you see, the promoter gene in this is provided with rho factor. This is for initiation of transcription. So, RNA polymerase will bind to promoter gene in presence of rho factor, and when it stops, the termination of RNA polymerase is with the help of sigma factor. So, rho factor to initiate transcription, sigma factor to terminate transcription. So, RNA polymerase, though it is only one, sigma factor and rho factor will guide them. So, after production of RNA molecule, it may be mRNA, rRNA or tRNA. This RNA will come outside that unwinded DNA loop. It is from 5' to 3'. This RNA molecule is said to be polycistronic. Now, this word we can use regarding to mRNA. Now, what is polycistronic? Single mRNA, M stands for messenger, single RNA carrying multiple messages. So it is called polycistronic. This is possible only in prokaryotes, never in eukaryotes. So as the messenger RNA is polycistronic, when it comes to the protoplasmic region, that is cytoplasm, ribosomes will bind to respective genetic messages. So as ribosome will move on the messenger RNA, rRNA will recognize the codons and tRNA will bring amino acids, and the amino acids are linked to form a polypeptide. So 1, 2, 3, like that multiple polypeptides, so called proteins, are synthesized on polycistronic RNA. So this process we call translation. This we are learning ahead. Today you need to understand what exactly transcription and how it occurs, who regulates the process in prokaryotes. Now, in eukaryotic transcription, as usual, the part of DNA unwinded, 5' to 3' shown in green color is the coding strand, and opposite complementary 3' to 5' green colored is a template strand, and the RNA production is shown in pink color from 5' to 3'. Here I have shown a dark line and dotted line. Means the message for protein synthesis. The message is there but not necessary for protein synthesis. Again a message for protein synthesis. So here the message is abrupted, not continuous. Now when this RNA molecule comes out, let us consider this is messenger RNA. When this comes out from unwinded DNA, it is called HN RNA. HN stands for heterogeneous RNA, as it shows non-coding introns. So dotted space is intron. Non-coding sequences, they are not at all involved in protein synthesis, and exons are coding sequences. Now here in eukaryotes, you should recall packaging of DNA. Eukaryotic DNA is always packed along with the octamers and forming nucleosome. Adjacent nucleosomes are connected to linker DNA. So DNA in eukaryotes appear like beaded string. So wherever the beads are there, those regions remain non-functional. So during transcription, when DNA unwinds, histone will go away and that region transcribe as intron. And linker DNA, which is having functional gene, that region transcribe as exons. So introns and exons are available on HN RNA. Now what next? The whole HN RNA is not necessary for protein synthesis. So from nucleoplasm, when HN RNA is about to enter in cytoplasm, it has to undergo splicing process. Splicing is a mechanism where introns are cleaved, means removed, and exons are joined. So there is a repair mechanism here. Removal of introns and joining of exons. This we call splicing. Enzyme necessary for splicing is spliceosome. These are nothing but proteins with the ribosomal units. Ribosome will see how the introns can be removed or non-coding regions can be removed. So during splicing, see these bubbles appear and the portion is cut with the help of spliceosome. After removal of intron, the RNA is functional now. It is messenger RNA from 5' to 3'. Now to this, these loops are removed. Imagine. So to this, capping starts. A protection is given for 5' end by adding methylated bases. It is guanosine monophosphate added continuously, giving a protection. So this specifies cap of RNA molecule. And towards 3' end, adenylated bases are added. It is called poly A tail. So capping, addition of methylated bases, and tailing, addition of adenylated bases. So splicing followed by capping and tailing. And this RNA molecule is functional messenger RNA ready to undergo translation. Now during translation, this messenger RNA attached to the ribosome will move from 5' to 3'. With the help of rRNA, they recognizes the messages. tRNA will bring one by one amino acids which are undergoing polymerization by synthesizing amide or peptide bond to form a protein or polypeptide. Only one protein here because this messenger RNA is monocistronic. Here it is polycistronic, here it is monocistronic. So now only you can compare what exactly happening in prokaryotic and eukaryotic transcription. With the help of video, I again explain separately transcription in prokaryotes and transcription in eukaryotes. Hope it is clear. Okay, you have to listen carefully and try to understand the similarity and differences in this transcription process. Today we are dealing with the most important topic for 5 marks. It is a transcription in prokaryotes. Already we discussed what is transcription. So let us see the bacterial cell having cell wall and the nucleoid inside. Nucleoid is having the DNA molecule supported by non-histones. Transcription unit is having a promoter region. It is a binding site for RNA polymerase with sigma factor. RNA polymerase and initiation by sigma factor unwinds the DNA of transcription region. A transcription complex is ready now. As RNA polymerase with sigma factor move ahead, the promoter region is recognizing the area for the transcription. Now the structural genes are open. On the template strand, you are observing addition of complementary nucleotides. Opposite is the coding strand. As there is addition of nucleotide, these are ribonucleotides. As they added, ribosome is connecting to the resulting RNA molecule and getting ready for further translation process. Once the transcription is over, there is a Rho factor which will bind to the resulting RNA molecule and ribozyme is released. RNA polymerase will detach and the process of transcription stops. So here RNA polymerase will continue the process of transcription till the terminator gene exposes on the template strand. This terminator gene is available on coding strand. When template strand towards which RNA polymerase reaches, Rho factor will come, binds to it and the process will stop. Thus, transcription in prokaryotic cell starts with initiation complex, continue as elongation, it is synthesis of RNA, and terminate on the terminator gene. So a transcription unit in prokaryote is continuous sequence of nucleotides. Promoter means it promotes transcription. Structural is the coded information having a genetic code transcribed on the RNA molecule. Only thing, they will replace the place of thymine by uracil. Deoxyribose sugar by ribose, and thus complementary nucleotides when they are linked on a template strand. When you compare that synthesized RNA and the coding strand of DNA, they are almost similar. Almost means 99%. Only in place of thymine you can see the uracil. Thus, a segment of DNA we call gene is regulating synthesis of RNA by exposing promoter region for binding of RNA polymerase with sigma factor to initiate the process, structural region to synthesize RNA molecule, and terminator region. When it is exposed, Rho factor will come, inhibits the activity of RNA polymerase enzyme, and the process of transcription stops. With this small session, I will conclude today. Remember, when the question comes for 5 marks, you need to explain first what is transcription. Give the transcription machinery components one below the other, diagrammatic representation, and then the mechanism. This is your answer for 5 marks. It is compulsory question expected in section D, part 1. With this, I'll conclude. Thank you very much.